So we're very happy we were able to attract Scott Baker to provide us with a keynote presentation today. Scott is a professor in the Department of Fisheries and Wildlife and Associate Director of the Marine Mammal Institute at Oregon State University. He did his undergraduate work at the New College of Florida, received a PhD in zoology right here from UH for his work on humpback whales, and he actually did a postdoc in molecular ecology at the National Cancer Institute and Smithsonian Institution. Before moving to Oregon State, Scott was a professor in the School of Biological Sciences at the University of Auckland. He has a Wikipedia page. He's one of the few people that I have met who have a Wikipedia page, where I discovered he was awarded the Bronze Medal in Science and Technology from the Royal Society of New Zealand for his work in applied conservation genetics. He also advised and participated in the award-winning documentary The Cove. His talk today is entitled Searching for Missing Whales with DNA.
All right. Well, thanks, Jeff, and thanks for the invitation to join you today, and to Aaron Olson, who nominated me, I think. It's always a pleasure to be back at the University of Hawaii and to see old friends and meet new friends, and to tell you a little bit about the work we've been involved in, one of which is I have a particular fondness for humpback whales in the North Pacific. A project that I've been involved in now for nearly 40 years, where we have new evidence of a missing breeding stock based on biopsy DNA. And this is an example of, I think, an application of conventional genetics or molecular ecology, DNA profiling, but to a large-scale database, an ocean basin-wide database, and a legacy database. That is a database that involves individuals of known identity from photo ID going back in time for now 30 or even 40 years, but also anticipated to continue to live for several more decades. So we're adding new individuals. Individuals are remaining in the population. New ones are coming in. Old ones are dying. And this is relevant to questions having to do with management issues for humpback whales in the North Pacific. I then want to switch topics a bit and tell you about some new work searching for new species of Baird's beaked whales with environmental DNA. And for this study, we've adopted genomic tools, in part to help compensate for really what is challenging sampling conditions for these kinds of species, and to help confirm or further validate newly recognized taxonomic unit. So just a little reminder about the impact of whaling in recent recovery. Those of us who've been involved in the study for a few decades have actually lived through a period of when humpback whale and other populations are actually recovering from pretty intense exploitation during the 20th century. So we know now that about 26,000 humpback whales were killed prior to IWC protection in 1966. And this now includes what we think are the complete records, including the Soviet illegal takes that the world was unaware of until following the dissolution of the Soviet Union. So now, taking that into account, we think oceanic abundance was reduced to less than 1,000 individuals ocean-wide, and now estimated to be more than 22,000, so a substantial recovery. In some ways, we know more about abundance, at least at the oceanic level, than we do about population structure, and that's partly because humpback whales have a complex population structure with seasonal migration and maternal fidelity to both breeding grounds and feeding grounds. So we see here a kind of a schematic of what we imagine the population structure to be, based primarily on photo identifications. This came out of a large-scale program referred to as SPLASH, Structures of Populations, Level of Abundance and Status of Humpback Whales, from 2004 to 2006. And they worked very hard to get that acronym, it's obvious. This involves sampling on all known breeding grounds and essentially all known feeding grounds at that time. Although, like all studies of that magnitude, they raise more questions. They raise as many questions as they answered, so now we have new evidence for new breeding grounds and feeding grounds. It was also a large-scale collection of genetic samples as well as photo ID. My laboratory was responsible for the DNA, the analysis of the genetic samples, the biopsy samples, and we've done this by a conventional DNA profiling that's fairly commonly used for individual identification of a number of cetaceans, not just humpback whales. So we have biopsy collection, DNA extraction, and then amplification and sequencing of mitochondrial DNA, and amplification and sizing of nuclear DNA in the form of microsatellite genotypes. So I'll focus mostly today on the control region haplotypes, actually in both components of the talk, because as a marker, it's highly informative where populations or species have these strong maternal fidelity. We also, of course, identify the sex of individuals. So interpreting mitochondrial haplotypes. Typically, we identify differences in mitochondrial DNA based on what's referred to as conventional Sanger sequences. This may be single base pairs or multiple base pairs. We typically identify these as individual haplotypes and reflect them or interpret them in the form of these pie diagrams. So we see differences in what we call frequencies of shared haplotypes in these two populations, and this would suggest a degree of reproductive isolation, or at least population differentiation. Now, this doesn't tell us about the difference or mutational or evolutionary difference of the haplotype, so we can reflect that in a dendrogram, often with these little bars reflecting individual variations or single nucleotide polymorphisms. So as I mentioned, mitochondrial DNA reflects maternal descent. Individuals of direct maternal descent will share mitochondrial haplotypes. Maternal fidelity will influence the distribution of haplotypes, reflecting cultural inheritance of migratory destinations. And we see significant differences in haplotype frequencies or indications of population structures and structure, and so used to define stocks or units to conserve, or even distinct population segments under the U.S. Endangered Species Act. So as a quick overview of oceanic mitochondrial diversity for humpback whales in the North Pacific, we can see that there are 28 different haplotypes reflected in this pie diagram. Most are rare. 4 are quite common, and it's really differences in the frequency of these 4 that drives primarily the differentiation of populations. We also see that the majority of the haplotypes are actually closely connected to each other. They're differing only by 1 or 2 base pairs. But there is a distinct group of haplotypes that are quite different at the evolutionary level and appear to reflect a historical colonization of the North Pacific by whales from the southern hemisphere. So using the SPLASH data set of nearly 1,000 individuals, 825, we were able to describe population differences between these primary breeding grounds, which are, of course, in some cases, geographically discrete or isolated from each other. In other cases, somewhat more complex. So we have evidence of differentiation between whales who winter here in Central America, whales that winter here in Mexico, whales in Hawaii, whales in Ogasawara, and whales in Okinawa, or the Ryukyu Islands, and the Philippines. So significant differences at all 5 of those differentiations. Feeding ground differentiation is a little harder to define because there's no natural barriers. There are, however, differences in the distribution and gaps in the distribution, and over the years, these have been used to classify these feeding grounds. And we see here differentiation of haplotypes as well, reflecting this maternal fidelity, but we don't see a one-to-one correspondence of a feeding ground to breeding ground, but in alternately a form of mixing of the breeding grounds on the feeding grounds. As I mentioned, SPLASH raised as many questions, or not as many, but raised questions as well as answering them. And one of those questions had to do with continuing uncertainty about the relationship of whales in the Russian Far East and what appeared to be a missing breeding ground, that is evidence from capture-recapture of individuals on photo identification that reflected a change in the resighting probability of whales in the Western Pacific. Currently, this was raised, in fact, by the Biological Review Team as a possibility of a 5th distinct population unit, but the NOAA Office of Protected Resources chose to take the available information and to recognize 4 distinct population segments, one of which is considered endangered, Central America, 2 of which are considered threatened, Mexico, and Asia, one of which has now been, as you know, most of you would know, no longer listed. That is, the Hawaii DPS is considered recovered. So in the last few years, there's been continued research on segments of these populations, particularly those that were maybe not as well represented in the SPLASH program. And we have worked with researchers from Russia and France on a collection of samples from the Keringinsky Gulf and from the Commander Islands. An interesting thing here is what appears to be a very distinct delineation of haplotype frequencies or population differentiation across a very fine scale. And they've interpreted this, and I believe correctly, as a cultural transmission of a fine scale fidelity to these different breeding sites. And so you can see, you know, a high FST, a highly significant difference in these haplotype frequencies. Along with this new information has come a closer look at the Marianas Islands by the team here from the Pacific Islands Fisheries Science Center, who've been involved in surveys of the Marianas Islands from 2015 to 2018, and have accumulated a body of evidence that implicates the Marianas Islands as a location of the missing breeding ground that was identified by inference from the SPLASH data. And this information has been synthesized in a publication that was just accepted for publication just this week, and congratulations to Marie Hill and Aaron Olson and their team for gathering all that information. I'll only speak to the genetic component of that analysis. So during those 4 years of surveys, they were able to collect 23 samples reflecting very strong differences between whales here in the Marianas Islands and those in the Ryukyu Islands, suggesting that there are two of these distinct population segments in the western North Pacific, one more closely tied to the Ryukyu Islands and one more closely tied to the Marianas Islands. The Marianas Islands also reflects an identity with those whales recently sampled and analyzed from the Commander Islands, whereas we see a greater identity of the Keringinsky whales with the Ryukyu or Okinawa samples. So now we have evidence of a DPS, we have evidence of at least what we think might be its primary feeding ground. And I know the Office of Protected Resources at NOAA is going to be very eager to revisit their biological opinion and designate an additional distinct population segment. So I'd like to switch topics now, or switch tacks a bit, and talk to you about searching for a new species of Baird's beaked whale with environmental DNA. And this is work that's being conducted with a number of collaborators, but in particular with Olga Filatova, Pew Marine Conservation Fellow and a faculty at Moscow State University. Now, just spoiler alert, I have used the word searching for a new species of DNA because we are still in the process of that search. But we have made some progress. So I think most of you would recognize that surveying cetaceans is expensive. The SPLASH program required about $2.6 million in field costs alone. Operating cost of a NOAA vessel are somewhere in the range of $35,000 to $60,000 a day. So any of these emerging tools that we can use to supplement or complement, you know, the conventional or traditional methods for studying cetaceans seem to be worthy of pursuit. I would also argue that species identification at sea is uncertain. It certainly is for me, even though I've been around cetaceans for nearly 40 years. There are 89 plus recognized species, not all of which are easily identified at sea. And of course, there are many stocks and distinct population segments that don't have conspicuous morphological characters. To me, confident identification requires a DNA barcode, using methods that we first developed for our surveys of whale meat markets in Japan and Korea, where we were concerned about the identity of whales and the potential exploitation or sale of meat from protected species. This involved both standardizing a particular locus, the mitochondrial DNA control region, which is sufficiently conserved to be amplified across all of the cetaceans, but sufficiently variable to give us information on identification of all known species. And we know this from accumulating a reference database. So we have a reference database of multiple sequences representing all 89 of the all recognized taxa until maybe last year or two, when there have been a few new recognized taxa. And this web-based identification program includes both control region and cytochrome B sequences sufficient for species identification. So we have the tools for identification based on a DNA a barcode of a kind, but do we have the sample? I mean, even with the greatest current technology, we need a little whiff of DNA. Well, obviously, there's a sort of a hierarchy of these sample availabilities where a stranded animal, the full carcass, or scientific whaling, the Japanese might argue, is the most efficient way to collect a genetic sample. But probably the gold standard is the biopsy dart, which takes advantage of medieval technology, the crossbow. And so as a biopsy darter, you are an arbalist, I've been told. There's also fecal sampling. Beaked whales are a particular challenge and a particular interest. There are 24 species as of 2019, the most recently just accepted based on a recent publication. 4 of these species have been described in just the last decade. These species are rare, elusive, or cryptic, or all 3. Some have never been seen alive, and most have poorly described, if any, information about population structure. And they have limited morphological characters, primarily the shape and position of the teeth, really like 2 tusks in the lower jaw, only in the mature males. And they are susceptible to Navy sonar. So there is a history of impact of these whales depending on naval activity, particular mid-range sonar. So what about environmental DNA? This review or perspective appeared just last year. Ellen Pickett was overviewing the status of environmental DNA and its potential for allowing us to identify relatively rare marine species. This is a methodology that's been pretty widely accepted in the freshwater aquatic system, where it's been used for identification of rare, for example, amphibians, as well as introduced species, but less applied, at least in the terms of the rare species approach to the marine environment. So the last several years, we've been involved in characterizing and optimizing environmental DNA detection and identification of cetacean species. The first phase of this involved what I guess I'd call positives, that is inshore sampling of killer whales from their path. So we knew that there had been a positive in terms of the whale's passage, and we were looking for a corresponding positive from the eDNA. And we used a new technology referred to as droplet digital PCR that I'll explain in a minute, as well as our conventional DNA barcoding. And now we've moved into the second phase of this work, open ocean sampling of beaked whales with environmental DNA and metabarcoding. So the first phase has now been published, the second phase is in progress. So a little more about this, and I appreciate the information of the previous, of Erica and Peter in introducing you to some of the methodology. But we've typically collected 2 liters of water, which we filtered through a 0.6 micron. We store that in the field with a Longmire buffer. We then extract it back in the laboratory, and we follow 2 paths of quantification and identification. One with droplet digital PCR, which is a methodology that takes a standard PCR reaction and fractionates it into tens of thousands of droplets using an oil emulsion, so that each of the droplets is an individual PCR reaction. Each droplet is then read by a flow cytometer and either indicated as a positive for the target DNA or a negative. If your DNA is very rare, then the presence or absence of the DNA in those droplets will conform to a Poisson rare event distribution. And the software then uses that rare event distribution to tell you how many copies of DNA you have in your sample. So this is similar to quantitative DNA, but doesn't require a standard, and is thought to be less sensitive to inhibitors and perhaps a bit more sensitive to very low copy number DNA. The second methodology that we've employed is now fairly standard to what next generation sequencing, where we recover or harvest the amplified DNA from the droplet digital PCR and submit it for sequencing on a MiSeq or similar, where we have a 250 base pair paired in reads so that the 2 fragments of amplified DNA, the two reads will form a contig that covers our entire 360 base pair target haplotype. We then use the program CHIME as a pipeline for merging these the paired-in reads and for identifying the distinct haplotypes. We finally submit these to the DNA surveillance website or to GenBank or to both. We're involved in a number of regional collaboration, mostly having to do with the presence of these enigmatic species, the beaked whales, some of which also involve naval activities. So we have collaborations on the Score Range in Southern California, and we have collaborations on the Autec Range in the Bahamas. So these are naval activity ranges that are in some cases are also equipped with hydrophone arrays for identification and localization of echolocating species. We're also involved in a study of a new, what may be a new species of beaked whale that's been identified acoustically by Erin Olson and her team. We refer to this as a deep water project because it involves DNA collected from the filtering of seawater from a deep water CTD cast. So open ocean sampling in the proximity of Beard's beaked whales, we've had the most success with this project. It's of particular interest because of a recent identification or recognition of a new species of beaked whale. So Baird's beaked whale are conventionally this gray form, but Japanese whalers and several recent strandings have provided evidence of a second form, or black form referred to by Japanese whalers as the karasu, which I think means crow or raven in Japanese. This is morphologically distinct if you see them sort of essentially next to a gray form, or so I'm told, but genetically, diagnostically different from the gray form. So the Baird's beaked whale's sister group is thought to be the Arnaud's whale, but now we have a third member of this genus, the Berardius minimus, that just published this year. So notice that these are the haplotypes associated with the gray form. These are the haplotypes associated with the black form. These are diagnostic differences in the nucleotide sequences of these haplotypes relative to their taxonomy. Note also there's relatively low haplotype diversity. Some haplotypes are relatively widespread, as indicated by these coloration pie diagrams. Some appear to be indicative of stock differences. But there are only 8 of these known or described or published in GenBank for the gray and only 5 for the black form. Now this black form has an interesting distribution as well, whereas the gray form is distributed pretty much around the Pacific Rim from Hawaii, from Japan all the way down to the Gulf of California. The black form has only been recognized from 8 specimens, 5 of which were here in the Aleutian Islands, and then the other 3 here off the island of Hokkaido. So one imagines that that is an artificial gap due to sampling or the absence of sampling. But we don't know that with certainty, and so this was one of our objectives, was to work with Olga on her study here in the Commander Islands, where we knew there were gray form, and to see if we could find evidence of the black form. So we want this to be an ecological tool. So we've developed a methodology of essentially trying to equip people in the field with the ability to collect the sample, filter the sample, and store it in some way, then bring it back to the laboratory. We can get as far as the filtering and the storage in the field. Obviously, the more technical aspect has to be done back at the wet lab. So Olga and her partner were very successful with this, having collected 53 surface samples during, I think, 11 different encounters with beaked whales. We were successful at finding beaked whale eDNA in all of these samples, the majority of which reflected this G1 haplotype. So out of a pool of all the samples collected in the Commander Island, all appeared to be the gray form, the majority of which were a G1 haplotype, the most common haplotype at least available in the current evidence, as well as a minority of G5 haplotypes. Interestingly, though, if we looked at the samples pooled only into days, that is, during which a group of individuals might have been sampled multiple times, we see significant differences in the haplotype frequencies, where again, G1 is the dominant or most common, but in some cases there's a substantial percentage of a G5 haplotype. In other cases, there are fixed differences, that is, there were no G5 haplotypes, perhaps reflecting a fine scale local population structure or social organization. So our conclusion from this and from our other applications is that environmental DNA represents a powerful new tool for detection and identification of cetacean species and stock structure. It now adds to this hierarchy of methodology for genetic sampling, particularly potential, I think has great potential for these cryptic species. I would note that just as a practical biologist, it gets harder and harder as you go along this gradient in terms of the laboratory methodologies and the extent of controls that you need to add to make sure that you're not sequencing artifacts. And where to now? Well, we need to continue to assess variability of eDNA, particularly for this type of application. Need to know a little more about the sources of environmental DNA with whales, its blows, feces, slough skin. Need to know a bit more about dilution and spreading if we're going to try to use these for occupancy models, for example. We need to know about rates of degradation accompanying those dilutions and spreading. Now, we're not going to get all that information in the near future, but I think there's sufficient evidence, and you've heard evidence already, that this is a methodology worth pursuing, so we need to think about how to integrate it into our more conventional methodologies for surveys of cetaceans. So whether we're going to undertake spatial or focal sampling, or whether we're going to sample at depth or at the surface, these are not exclusive. These can be, we can do both. Whether we're going to be aided by visual or acoustic detection, and ultimately or eventually whether we're going to be able to have onboard DNA sequencing so that we can transfer more and more of the chain of information to the field itself. And with that, I'll say thank you again for the invitation.
We have time for questions? No?
Now, certainly Brian Bowen has a question.
Okay. Oh, go ahead, Brian.
Are there other frontiers in cetacean research where you think environmental DNA is going to recover more biodiversity?
Well, I think there's the potential to sample large social groups much more efficiently with eDNA, for example. I mean, you find a pod of 400 Pepanocephala, which is a species we know from the coast of Hawaii. You're not going to be able to collect 500 or 400 biopsy samples to reflect the diversity of mitochondrial DNA, but I think a relatively small number of eDNA samples collected right from the path or the wake of that population would give you potentially the diversity, maybe not quantitative, although not excluding that. So I think there's the potential, as we found with the Baird's beaked whale, to get a reflection of multiple haplotypes and perhaps something about the frequency of those. And that could be used as a kind of a shorthand for comparisons to other populations to look for population differentiation. And there's been some work on that already. Kim Parsons published recently an article on harbor porpoise from Southeast Alaska, which have been impossible to sample with a biopsy, so that the only available samples came from bycatch or beach cast individuals. And those are almost certainly not really representative of populations. So she's been able to use eDNA to uncover considerably more structure than had been apparent from those previous samples. So I think we've got a lot of potential. We've just got to find a fit of the method to the question and the problem.
Do you have a difference in what you can learn from populations that have gone through a genetic bottleneck, where you were saying humpbacks went down to 1,000, number of populations?
Yeah. Right. The question is about bottleneck effects. There is evidence for bottleneck effects for a few of the really intensively exploited populations. We see a reduced diversity of mitochondrial DNA in humpback whales in the North Pacific, but I think that's an evolutionary consequence of their demographic history. I say that because we see those many haplotypes differing by only one nucleotide. And that's a pattern that suggests a recent expansion of a population, not a pruning of those, which you might expect in a bottleneck. A species like the Antarctic blue whale, which was probably numbered 300,000 before 20th century exploitation, was reduced to numbers that were almost impossible to estimate. They were so low that there was an estimate of 400, but it was 400 plus or minus 200. A species like that, I think there is some evidence based on analysis of DNA from bones of historical records that there's been a loss of mitochondrial diversity for certain.
Last question.
Do you have a sense of the temporal and spatial scale that are associated with your environmental DNA sampling? What I mean is how far away
Yeah.
the whale must
Right.
be and how long ago should be depth water for you to be able to detect it?
Yeah, my impression based on our results to date is that the spatial distribution of the eDNA is highly variable from the whales and very heterogeneous. So you could sample on the left side of the boat and the right side, and the left side might have a positive, the right side might not. You might wait around an hour and do that again, and one or both of them might have it again, but not necessarily consistently. Temporal aspects, we've experienced. I think that the degradation problem is not as great as probably I would have thought before I started this work. I thought that would be the primary limiting. Now I think it's more the spatial heterogeneity component, and that the DNA is probably a bit more persistent in most environments. Of course, it's going to be temperature dependent, light dependent, and so forth. But it's probably a bit more persistent than we might have thought, and maybe less degraded. Partly because a lot of it may still be attached to particles. It may still be in the mitochondria when we filter it out. So it's been supercoiled and somewhat protected.
Okay, thanks, Scott.
Yeah, thank you.